Welcome to my talk for the APS March meeting. So I'll be talking on the spatial segregation and organization of bacterial chromosomes. So before I explain our results, I'll give a brief introduction to define our question and then move ahead. So as we know, bacteria reproduce and and there's cell division that occurs in bacteria. But just before cell division, the chromosomes must make a copy of themselves, and this process is known as replication. So the replication has to be carried out faithfully, and these replicated chromosomes must spatially segregate before the occurrence of cell division. Now, in higher order organisms such as eukaryotes, there's already a dedicated machinery for this, which are known as mitotic spindles. However, bacterial chromosomes lack any such dedicated machinery, and especially primitive ones like E. coli, they lack a dedicated protein based machinery to do this. Therefore, they must rely on entropy to accomplish the segregation of its chromosome. This was first suggested in 2010 by Subjun and others. Uh, the key idea is they suggested that newly replicated chromosomes, as you can see on the right, newly replicated chromosomes are seen in the cell, these chromosomes can spatially segregate due to entropic repulsion between the daughter chromosomes. The key idea is that the E. coli chromosome being a ring polymer, so now there are two ring polymers, and if they spatially segregate, they have access to more number of conformations, and as a result, their conformational entropy is higher if they spatially segregate. So therefore, there's an effective repulsion between the two daughter chromosome ring polymers, which is purely entropic in origin. However, this entropic model can't explain several things, and the chief among them is the so-called loci localization experiment. So this is an experiment which was done by the Wiggins group in 2016. Here they tagged several loci or points along the chromosome contour, and they have fluorescently tagged them so they can follow the trajectory of these points as a function of time. So if you can see my cursor, I'm pointing to the ODC, which is one of the locus. And the ODC starts out from the mid cell position, mid cell with respect to the cell long axis. And then the ODC is the first locus to get replicated. So now there are two ODCs after a while. So the initial ODC branches out from the mid cell position, and then it gets replicated, and the two ODCs move to the quarter positions along the cell long axis. So if you can follow my cursor, I'm pointing to the mid cell position, and this is with respect to the cell long axis, and then the two ODCs eventually move to the quarters, that is with respect to the cell long axis as well. Similarly, the other loci along the chain also get localized to certain cellular addresses, and as you can see here, the other loci, TER1, R3, L2, and so on. The question is, how do these loci get localized and why? And surely it is incompatible with the entropy model as it is, because the entropy model would predict that the loci can be present anywhere along the cell long axis. It need not be localized to certain points along the cell long axis. However, the question is, can the entropy model be modified suitably so as to obtain such unique localization patterns for these loci? The answer is yes. And the key insight that we offer is that we take a polymer model of 500 monomers, and it's a ring polymer, but now we modify the ring polymer suitably such that one has three loops, such that the ring polymer now has an internal looped structure as well. So we cross-link certain monomers along the chain. For example, here we cross-link monomer 1 and 125 and 1 and 375. And monomer number 1 is, by the way, connected to monomer number 500 because it's a ring polymer. And we denote ODC as monomer number 1 and the TER as monomer number 250. So using this mechanism of cross-linking, one ends up with two small loops and a bigger loop. And this looped architecture will have consequences with regards to loci localization. But the question is, are these cross-linking sites viable biologically? The answer is yes, because these effective cross-links may be realized by SMC proteins, which are known to link distant chromosomal regions in space. And moreover, these effective cross-links may also arise due to the co-localization of ribosomal RNA. And in fact, we provide the references here. In fact, in this genes and development paper, we found out that the location of these RRN operons closely match that of the cross-linking sites that we have chosen. So for our study, we use Monte Carlo simulations, and the chromosome is represented as a bead spring ring polymer, of course modified suitably. It is confined to a cylinder of aspect ratio 1 is to 2.5, and we maintain a volume fraction of 0.2. These are all parameters which are seen, which is experimentally measured for the E. coli chromosome. And since we take a 500 monomer chain, each bead represents 9.2 kilobase pairs of DNA. So the two ARC2 polymer, the polymer is in the ARC2 architecture to begin with, as you can see on the right, and then it's confined to a cylinder, and we model replication explicitly. So after the process of replication, one has two ARC2 polymers cross-linked at the TER, and the cylinder also grows in accordance to what is known. The cylinder grows as replication proceeds and grows to twice its initial length. More importantly, we also allow chain crossing at infrequent intervals by reducing the bead size of a diameters. This is done to mimic the effect of topoisomerase, which is present in the cells, which allows the release of topological constraints. Using our model, we find that we obtain the localization patterns of the loci. For example, here we show the localization for ODC, which is shown in red, and it is nicely peaked around the mid cell position. This is for a single ARC2 polymer, whereas for ARC0, which is just a ring polymer, we see that there is no such localization pattern. Similarly, for the replicated chromosome case, we have two ODCs which get localized to the quarters, while ARC0, which is just a ring polymer, doesn't show any localization pattern again. So ARC2 successfully localizes these loci, and this is true for other loci as well. But the question is, why and how does this happen? So now to answer that question, we would like to look at ARC2 in detail. Here we look at the loops of ARC2, and they're colored differently. The two smaller loops are colored pink and green respectively, and the bigger loop is shown in blue. And we follow the distribution of the center of masses of these loops. And this is for the two ARC2 polymer case, or the replicated case. We follow the distribution of the center of masses, and we see that the distribution for the two smaller loops, the distribution of the center of mass of the two smaller loops, shows very little overlap with the distribution of the center of mass of the bigger loop. And that is true for the two polymer case as well as the single polymer case on the right. We see that the distribution of the two smaller loops shows very little overlap with the distribution of the bigger loop, which indicates that the two smaller loops entropically repel the bigger loop. Now, ODC, which happens to be the junction of these loops, get localized to the quarters and the mid-cell position respectively, but other loci, which could be situated in any of these loops, are confined to particular regions along the cylindrical long axis of the cell long axis. Now we already know that the E. coli chromosome is known to have a macrodomain-like organization. Uh, However, ARC2 fails to give us the macrodomain-like organization, but if we modify ARC2 suitably such that there are two additional smaller loops, and we call it the ARC2-2 architecture, such an architecture gives rise to the macrodomain-like organization. as we show in the next slide. Here on the left, I show experimental data from Leoy and others from 2018. And this Hi-C data shows which parts of the chromosome are in close proximity to which other parts. We find that there are bright patches which are roughly 1 megabase pair in size, and they correspond to what are known as macrodomains. We find that in a simulated contact map shown on the right, we have similar bright patches of roughly 1 megabase pair size, and therefore we say that we have recovered the so-called macrodomain organization of the E. coli chromosome. But however, there are still further questions like what kind of polymer architectures lead to faster segregation? Could other polymer architectures lead to localization of loci? And could other polymer architectures be relevant to other bacterial chromosomes? To answer these questions, we have done a detailed investigative study using 12 different architectures, and these architectures could have a linear looped arrangement or a side looped arrangement. And we found out that one of the investigated polymer architecture, the so-called ARC9, shows a striking resemblance to the localization data for the C. crescentus bacterial chromosome. On the left, I show data from the experiments conducted by the Lucy Shapiro group in 2004, where they have done fluorescence experiments and looked at the localization of certain loci, and we recover those localization patterns for the same loci using our ARC9 architecture, as I show on the right. Furthermore, the Hi-C data for C. crescentus also shows a match with our simulated data. Here we show data of the experiment conducted by Ley and others in 2013. So in the C. crescentus Hi-C map, one has the double diagonal Hi-C feature, the double diagonal feature. We recover the double diagonal feature in our simulations as well. So the Hi-C data also shows a relatively good match. Therefore, we believe that a polymer architecture plays a key role in the organization of bacterial chromosomes. And for more details and insights regarding our work, the reader is suggested to refer to these preprints that we have put out. Thank you.